in RNA-seq data, you will always see duplication. So in RNA, you have housekeeping genes like actin, tubulin, malate, these things. So genes that are expressed in very high levels in the cells. And there are millions of transcripts for these genes in a cell. And transcripts are not that long. Compared to a genome sequence, they're quite short. So if you're going to sonicate them, and you have millions of identical transcripts there, even with a random process like sonication, you will generate the same fragments. So this assumption doesn't apply to RNA-seq data. And these housekeeping genes will generate copies, identical copies. So in RNA-seq data, you always see duplicates, and part of these duplicates will be coming from highly expressed genes. And these copies are real, and this is very important. They are biologically real because they come from different transcripts from the same gene. Yeah? They are biologically real, so you want to keep them in your data, and in a later stage, you want to count them because they represent different transcripts. They are biological copies. Okay. So that's one reason why you see in RNA-seq data duplication. Another reason why you see duplication in RNA-seq data is the ribosomal RNA. Although you try to remove it as much as possible, there still will be some ribosomal RNA in your RNA libraries. And ribosomal RNAs are very similar, so they will generate identical copies. That's another reason. And indeed, this peak here at the utmost right of your plot, in almost all cases, indicates something that is called adapter contamination. Where does that come from? Well, during your library preparation, you create your fragments by sonication, and then to these fragments, you're going to add adapters. So these are small pieces of DNA that contain, among others, sequences that will bind to the sequencing primer. Yeah? So pieces of DNA that you need for sequencing. And you're going to ligate these adapters to your fragments. Now, ligation is a process that you really can't control. You put everything together with a ligase enzyme, and you hope that it works out. And it will work out, but if you have a lot of adapters and you don't have that many fragments, what will happen is that the adapters will bind to each other also. They will bind to your fragments, but they will also bind to each other. So in your library, you will have what is called adapter dimers. So small fragments that consist completely of adapter sequences that were ligated to each other. But since they contain the sequencing primer, they will be sequenced along with your fragments, and they will end up in your FASTQ file. And when they are there, they typically pop up here at the utmost right of the plot, because it will be one sequence, the adapter sequence, one sequence that occurs a lot in your FASTQ file. And that's what you see here, or here. So a sequence that occurs very, very, very often. Your housekeeping genes and your ribosomal RNAs will typically end up here in this region. So these peaks that you see here are probably coming from housekeeping genes. This one is very likely adapter dimers. Now, apart from what I already explained here, in the lowly expressed copies, you can also have what is called technical duplicates. Technical duplicates are generated by PCR. So in your library preparation, there's a lot of steps that involve PCR. PCR will generate copies. Yeah, but these copies are not biologically real. Yeah, they are not. They are copies that are coming from exactly the same transcript, from one transcript. So you don't want to count these as copies. You don't want to see them as copies. They're not biologically real. What is the annoying thing in RNA-seq data? You cannot make the distinction between your biological copies, these ones, and your technical copies that are generated by the PCR. There's no way to distinguish them unless you use UMIs. But most people don't. So you cannot distinguish them. What should you do in that case? Well, the assumption is that in RNA-seq libraries, most of the copies that you see are coming from housekeeping genes. So they are biologically relevant. So what they do is they keep all the copies in. Yeah, every one of them. And yes, you will have some noise because of this, but there's nothing you can do about this. Okay. So, but this is indeed why we see duplication. So, and in this plot, what you look at is, do I see this peak here? Yeah? Do I see a peak at the utmost right? If you see a peak at the utmost right, it can be adapter contamination. For RNA-seq data, seeing peaks here doesn't matter. Yeah? He will never be happy when you give him RNA-seq data because there will always be duplication here. But that duplication is fine. Most of it is biologically relevant.